Good morning, students. Today we are going to learn about disorders of skeletal muscles. In that, we will learn about denervation, hypersensitivity, and fibrillations in skeletal muscle. First, coming to denervation hypersensitivity, the learning objectives are definition, history, causes of denervation, denervation hypersensitivity seen in which tissues, molecular mechanisms involved, changes in the neuron after its injury, and changes occurring in the skeletal muscle after denervation and after reinnervation. And denervation hypersensitivity in smooth muscle and exocrine glands. And applied physiology and recent advances we will see. In 1939, he proposed law of denervation. So what it says? When in a series of efferent neurons, a unit is destroyed, an increased irritability to chemical agents develops in the isolated structure or structures, the effect being maximal in the part directly denervated. So he formulated the law of denervation canon in 1939. So according to this, when a series of efferent neurons of a unit is destroyed, an increased irritability to the chemical agents develops in the isolated structures. So the effect where it is maximum is the part which is directly denervated. And Claude Bernard gave an extension to the law of denervation. Okay. And in 1971 also, Lunt, Stephanie and D. Robertis, all these scientists have worked on this denervation hypersensitivity. The causes of denervation are because of injury. There may be neuropraxia or axonotmesis or neuroatmesis, or diseases like polio, post-polio syndrome, and ALS, that is amyotrophic lateral sclerosis, and surgical procedures like sympathectomy and vagotomy all will lead to cut in the nerve supply to the skeletal muscle. Denervation hypersensitivity is seen in skeletal muscles, smooth muscles, and exocrine glands except sweat glands. The molecular mechanisms which are involved are upregulation of the receptor in the end organs, that is, receptors which are present on the motor end plate, they are increased, and loss of mechanisms for transmitter removal. So when acetylcholine is released in the synaptic left, there is no acetylcholinesterase at that level. So there is loss of mechanism for removal of the neurotransmitter. And there is increased post-junctional responsiveness. And there is altered synthesis of subunit receptors. So all these are the mechanisms involved in the denervation hypersensitivity. See, this is the changes. These are the changes in neuron after injury. That is Valerian degeneration. So this has this all these things. Okay. So what happens within 24 hours of injury? Okay, after 24 hours to 48 hours of injury, and 72 hours of injury, and after 1 week of injury, what happens? We all know this. So there is a separate video on Valerian degeneration. Now we will see the changes in skeletal muscle fiber after denervation. So in skeletal muscle fiber, we can see fibrillation potentials. They will appear after 1 week of denervation. They are the spontaneous repetitive action potentials. They are formed because of increased acetylcholine receptor over the entire muscle membrane surface. Actually, the acetylcholine receptors, they are concentrated at the motor end plate. But now what happened? These receptors are present everywhere on the muscle membrane surface. So that is one cause of the fibrillation potentials. And these are recorded by electromyography test or NCS test, nerve conduction study test, and they will persist until the muscle fiber get re-innervated or until the muscle fiber becomes necrotic. Coming to skeletal muscle atrophy. What is meant by atrophy? Atrophy means decrease in muscle fiber diameter, and there will be increased concentration of the myonuclei, and there is breakdown of the collagen. It is the early change which occurs in the denervation atrophy. Okay. So if you stain with the para-iodic acid skiff, the staining will be faint or absent. And there is hypertrophy of the normal innervated muscle fiber due to increased workload on that. See this is a denervated muscle fiber. It is the intracellular area and this is the extracellular area. Now there is no signal, so there is no work for the muscle fiber. There is mechanical unloading. See this is the cell membrane. So due to this is the putative stimulation. So due to this, calcium will be more in the cell, and reactive oxygen species also increased inside the cell. And these reactive oxygen species will stimulate the insulin resistance. That is, it will impair the IGF-1 signaling. And these reactive oxygen species will also cause calcium overload in the cell. And this mechanical unloading also directly it will stimulate the insulin resistance. Now what happened? The muscle is going into atrophic conditions. Why it is going into atrophic conditions? So see here, the calcium overload condition, it will stimulate the calpains. So calpains will be increased. Normally these calpains will be inhibited by calpastatin, but here this calpastatin protein is decreased. Now because of increased calpains, the muscle undergoes atrophy. And increased reactive oxygen species is also a cause for increase in the calpains. And these reactive oxygen species also increases caspase 3 in the cell, and this also leads to muscle atrophy. And this caspase 3 is also leading to proteasomal proteolysis, and this proteolysis is resulted in muscle atrophy. And the reactive oxygen species will increase the nuclear factor kappa B in the cell, and it will increase the other proteins which will cause proteasomal proteolysis, eventually leading to muscle atrophy. And these impaired IGF-1 signaling, that is impaired signaling that is causing insulin resistance, is leading to formation of IP3K or PI3K protein. This will phosphorylate the AKT protein and it decreases the activity of the AKT protein. Normally, this AKT protein will inhibit the FOXO protein. Now, this FOXO protein is not inhibited, and the upregulation of the FOXO protein will be caused by decrease in the AKT protein and because of reactive oxygen species also. This is causing the activation of the enzymes which are causing proteolysis, and it also leads to increase in the genes which are related to autophagy. All these things are ultimately leading to atrophy of the skeletal muscle fiber. Now coming to the re-innervation of skeletal muscle fiber. So what happens if the denervated skeletal muscle fiber is re-innervated. So there is formation of particular type of muscle fibers only. We have different types of muscle fibers, that is type 1 and type 2. Now all muscle fibers are type 1 only. So mostly 99.9% only type 1 muscle fibers are formed, whichever may be the type of muscle fibers destroyed. So that is called fiber type grouping. It is the hallmark of denervation followed by renervation. So in a normal skeletal muscle, we have different types of muscle fibers. But if we are seeing only type 1 muscle fibers, that is the hallmark of denervation followed by renervation. This is called fiber type grouping. And also we can see type specific group atrophy. That is only one one group of type muscles are atrophied in the skeletal muscle. Now coming to the factors regulating re-innervation. See this, the expression of the MIR206 will be enhanced in the skeletal muscle fiber. So what this will cause? It will promote the regeneration of the neuromuscular junction after denervation. So this is helpful for the NMJ regeneration. And also this MIR206 will inhibit the HDAC4 protein. This HDAC4 protein will normally inhibit the fibroblastic growth factor, okay, FGF-BP1 protein. But now this inhibition is not there. Now this FGF-BP1 will bind to FGF, fibroblastic growth factor, and activates the FGF. This FGF is important for promoting the re-innervation, and there is marked proliferation of the acetylcholine receptors. So these are the factors which regulate the re-innervation. So the eccrine sweat glands, okay, these are the one type of sweat glands. They will present almost on the entire body surface. So these exocrine glands, they do not obey Cannon's law. And the recent advances are increased acetylcholine receptor mRNA is noted after uh, denervation, that is within 3 days after denervation. So the mRNA will increase up to 100-fold. And increased expression of HUR protein is noted. This HUR protein helps for the stabilization of the beta subunit of the acetylcholine receptor. And P38 MAP kinase pathway activated following denervation, it will activate the HU. It is a RNA binding protein human antigen. So these are the recent advances. Now coming to the pharmacological denervation hypersensitivity. Means what? It is also called pharmacological disuse hypersensitivity. That is after an exposure of an antagonistic drug, okay, for a long time, the muscle will show pharmacological denervation hypersensitivity. So there will be increase in the sensitivity by a postsynaptic cell because of decrease input by the incoming axons because of the usage of the antagonistic drug for a long time. Now coming to the second topic, that is fibrillations in skeletal muscle. So actually we will see fibrillations in the single muscle fiber. So they cannot be seen with a naked eye. Skeletal muscle fibrillation refers to small local muscular contractions that occurs in response to spontaneous activation of single muscle fibers, which then contract independently of surrounding fibers. So these muscle fibers will contract independently. This activity, in contrast to the fasciculations of the muscle, these fasciculations of the muscle occurs as the motor unit the entire muscle fibers supplied by one motor unit are contracted, and all those muscle fibers will contract in a synchronous manner. Spontaneous fibrillations have been described in cases of certain muscle disorders such as muscular dystrophy, polymyositis, and amyotrophic lateral sclerosis, and conditions in which muscle fiber regeneration or denervation are present. So, in all these conditions, we can see spontaneous fibrillations. These spontaneous fibrillations are often used clinically to determine the severity and magnitude of the neural injury and nerve regeneration status. So, these fibrillations we can use clinically for determining the severity of the neural injury and the magnitude of neural injury, and to know the regeneration of the nerve status. It also has been proposed that these fibrillations could be useful in assessing the muscle atrophy occurring subsequent to denervation also. So in all these stages, we can use these fibrillations present or not, and we can assess some clinical conditions. Coming to the history, the first recorded observations of the muscle fibrillations after denervation is by Schiff scientists. So he reported in his article in 1851 that this is the name of the article, that visual observations of fibrillations in the tongue muscles of dogs after bilateral hypoglossal nerve section. So there are many fibrillations coming in the tongue muscles of the dog. So he observed that in 1851. In 1915, experiments by Lansley and Cato, they were the first to show explicitly that fibrillations follow denervation. So they have proved that after denervation, the muscle fiber shows fibrillations. 3 years later, Lansley and Hashimoto scientists, they continued the experiments on this topic and have come close to observing length dependency phenomena. Okay, so what is this length dependency phenomena in denervation, that is in fibrillations? What is that is the currently accepted idea that the fibrillation potential appearance is dependent upon the distance of the muscle from the lesion. So the fibrillation potential, the fibrillations are depending upon the distance between the lesion in the nerve and the supplied muscle. So when the distance between the neural lesion and the muscle is smaller, then the potential will appear earlier. So the less distance, the potential will appear earlier. So the fibrillations will appear earlier. So these scientists experimentally sectioned the sciatic nerve of two groups of animals. So in one group they have cut the nerve just near the origin of the nerve, and in other group they have cut the nerve at the muscle end. So they have seen that the fibrillations started earlier in the second group, that is when the fiber is cut near to the muscle. So in first group what happened, they have cut the nerve at the beginning, so the entire axon will be undergoing into degenerate condition. So in those cases, so in that first case, the fibrillation potentials will appear very late. Now coming to origins and consequences of muscle fibrillation. When a muscle is severed from its nerve, what happens? The muscle loses both the electrical stimulus the nerve provided as well as any chemical neurotrophic factors the nerve would have supplied. So, it will lose two things. What are those two things? It will lose electrical stimulus which the nerve is providing, and it lose any chemical factors which the nerve is releasing. That is, those are neurotrophic factors. During 1940s to 50s, the scientists experimentally determined that the membrane resting potentials of denervated muscle fibers tended to oscillate. So the RMP of the denervated muscle fibers is not a constant. It will undergo oscillations. This was ultimately the important finding, as the membrane oscillations is one of the key concepts in etiology of fibrillation potentials. Over the years, researchers have investigated losses of both electrophysiological input as well as potential neurotrophic factors as possible origins of fibrillation potentials, and have provided numerous reports which ultimately demonstrate that it is mainly the loss of electrophysiological stimulation that is responsible for the changes occur in the sarcolemmal membrane and propagate the spontaneous fibrillations. So out of these two, the most important is electrophysiological input. In turn, the fibrillations themselves may affect the cellular metabolism and muscle fiber size. So first we will see the electrophysiological factors. Numerous studies report that changes in resting membrane potentials occur within a few days of denervation, concomitant with the onset of fibrillation potentials. So within a few days of denervation, the RMP will fluctuate and fibrillations will start. Purvis and Sackman reported in 1974 that spontaneous fibrillations arose from both rhythmically and irregularly discharging fibers. So they have observed that both rhythmically discharging fibers and irregular discharging fibers, they will gives rise to spontaneous fibrillations. So both types of events were inhibited when they treated with the TTX, that is tetrodotoxin. This is a sodium channel blocker. So they have worked on a rat diaphragm preparation, okay, and they have added sodium channel blocker tetrodotoxin. So they have seen that fibrillations have stopped. So what it implies? The researchers concluded that fibrillations in both types, that is rhythmically discharging fibers and irregular discharging fibers types, the fibrillations types arose from localized changes in the conductance of the sodium. The normal levels of the calcium are not sufficient to stabilize the muscle fiber membranes. These also lead to fibrillation potentials. Another explanation for the increase in the conductance of the sodium is there is increase in the ovabine binding site on the sodium potassium ATPase molecules on the cell membrane of the denervated muscle. So this suggests that there is increase in the sodium potassium ATPase. The Pepon scientist also reported the discovery of tetrodotoxin resistant sodium current. So up to now we are discussing sodium channel and sodium current, but they have discovered tetrodotoxin resistant sodium current. So expression of this tetrodotoxin resistant cardiac type of sodium channel has reported in the denervated muscle. And Sekiguchi and colleagues have recently reported that tetrodotoxin resistant channel that is expressed in denervated potential with the onset of fibrillation potential. So this is also a cause of fibrillations. They used a northern blot analysis technique to demonstrate that the tetrodotoxin resistant cardiac type voltage gated NAV 1.5 sodium channel mRNA is upregulated by second day after denervation. So northern blot analysis is for protein analysis. They have seen this mRNA protein after second day of denervation. So that is about sodium current. Now coming to the potassium current changes. There will be changes in the potassium currents and channel expression also in the denervated skeletal muscle membrane. There is decrease in potassium permeability reported to occur in the denervated muscle. And it is also suggested that this could alter the sodium to potassium permeability ratio and contributes to the decline in the membrane potential. And there is a disappearance of 81 ps voltage dependent potassium channel from the denervated muscle, and there will be appearance of 16 ps channel in the denervated muscle, which is similar to SK channel. What is this? The scientists performed patch clamp technique and described that small conductance, SK means small conductance potassium channel. So this is the calcium activated potassium current that is generated in the denervated mouse skeletal muscle, which was absent from the reinnervated, which is absent from the innervated controls. So there is formation of the small conductance potassium channel. And some studies also suggest that the changes in the cellular calcium levels or calcium handling by the cell may be involved in the modulation of the fibrillation potentials. Indeed, it has been shown that post-denervation, intracellular calcium levels will increase in the skeletal muscle, and the rate of calcium uptake in the sarcoplasmic reticulum declines. This will result in membrane depolarization. Studies treating denervated cats with dantrolene. What is this dantrolene? This is the inhibitor of the ryanodine receptor mediated calcium release from the sarcoplasmic reticulum. It reveals inhibition of the fibrillation activity. So, if you use dantrolene, it is decrease in the fibrillation activity. So, what you can confirm? The calcium is coming from the sarcoplasmic reticulum into the sarcoplasm and causing fibrillations. Now coming to the neurotrophic factors. The neurotrophic, the term has been used to refer the chemicals that are released from the neurons, and they will have a regulatory effect on the muscle physiology. Numerous studies have shown that an increase in the sensitivity to acetylcholine shortly follows denervation, and this increased acetylcholine sensitivity spreads from motor end plate to the surface of the muscle fiber also. The increased sensitivity is believed to be due to increased synthesis of acetylcholine receptors. So, in the muscle membrane, what is happening? Not only at the motor end plate, but all over the muscle fiber membrane, there will be formation of acetylcholine receptors. So, the sensitivity is also increased for acetylcholine. And there is autocrine activation of the nicotinic acetylcholine receptors, and this will increase the intracellular calcium levels and leads to contraction of the myotubules. Next, coming to the metabolic factors. Small changes in the temperature have a large direct effect on spontaneous fibrillation in denervated skeletal muscle. These data suggest that the level of spontaneous fibrillation activity is related to muscle metabolic rate. So, if the temperature is increased, the fibrillation activity is increased. Denervated muscle exhibits decreased levels of glycogen and changes in the activity levels of numerous enzymes like succinic dehydrogenase is decreased, and cytochrome oxidase is decreased, and hexokinase activity is increased, and other factors are altered which are involved in the carbohydrate metabolism. Increased phosphocreatine occurs and a drop in organic phosphate occurs within 24 hours. But after 3 days, what is happening? Organic phosphate was still low, but phosphocreatine is returned to normal. And ATP levels remain nearly constant for the first 5 days, but after that they will drop. The decreased resting membrane potential of denervated skeletal muscle would be brought about by the metabolic changes which deprived the tissue of adequate energy supplies, and this will disrupt the sodium potassium pump, because to maintain the sodium potassium pump we need ATP. Now ATP is low, and there is increase in the lactate production and increase in the lactate oxidation, and fibrillation rate, firing rate was positively correlated with temperature and partial pressure of oxygen. So this suggests that the rate of fibrillation potential initiation is proportional to the rate of aerobic metabolism. What are these satellite cells? The cells away from the denervated muscle. So these satellite cells will get activated and they will proliferate. That means they will increase in the number and they will fuse to myotubes or myotubules and grow into the regenerated muscle fibers. So from in vitro myogenesis, what they have noticed is the new fibers soon or later starts to contract and they may contribute to fibrillation of the denervated muscles, in particular of long-term denervated muscles. So fibrillations because of the satellite cells. Now coming to the fibrillation potentials and atrophy. In the early 19th century, it was suggested that the atrophy which occurs subsequent to denervation was a consequence of the excessive energy usage incurred by the spontaneous fibrillations, and so denervation induced atrophy was referred to as fatigue atrophy. So the muscles undergoing fatigue atrophy they have noticed, that is they have identified in the early 19th century. But in 1940 this theory was uh, re-examined and the researchers demonstrated that by inhibiting the fibrillation to the denervated muscle with the quinidine, okay, or quinine, the atrophy did not stop. Thus, it is accepted that the stimulation from fibrillation potentials actually counteracts the muscle loss to some extent. So, actually, fibrillation potentials are safe. So, the muscle won't undergo atrophy. Diagnostic value of fibrillations in peripheral nervous system injuries and disorders. Clinically, the presence or absence of fibrillation potentials is used to help to determine whether a nerve injury either axonal or demyelinating in nature. So these fibrillation potentials are easily detected by EMG and in general these are detected within 1 to 4 weeks after denervation. The variable time of onset is attributed to nerve degeneration being length dependent. So we have talked about length dependent mechanism and therefore fibrillation potentials are expected to occur earlier in muscles closer to the site of nerve injury than in muscles farther away. Next, coming to prognostic value of fibrillations in peripheral nervous system injuries. Fibrillation potential persistence varies with the species and with the distance of nerve lesion from the muscle. In animals, investigators have noted that the disappearance of fibrillations heralds the return of motor function after nerve injury, and it is useful to evaluate the timing of reinnervation. So, if the fibrillation stops, it will indicate the motor function is returning. Incremental denervation is among the mechanisms producing muscle atrophy in aging, that is sarcopenia. That is, in aging, the muscles undergo atrophy, it is called sarcopenia. So, why it is occurring? Because of incremental denervation. That is the mechanism involved in the aging persons. Indeed, it would be most interesting to confirm by longitudinal studies using single fiber EMG and dynamic echomyography that skeletal muscle of normal seniors experience incremental levels of subclinical denervation of muscle fibers. So in aging we can do SFEMG and dynamic echomyography to see the subclinical denervation of the muscle fibers. Next, coming to usefulness of fibrillations for monitoring therapeutic interventions. You see this Valerian degeneration. In this Valerian degeneration, it is a process whereby nerves degenerate after axonal injury, and it is characterized histologically by axonal blubbing and fragmentation into ovoids. So the clinical correlation of Valerian degeneration is a failure of electrical conduction by the nerve distal to the site of injury and the onset of denervation potentials. In turn, this information is used to prognosticate the time course and the extent of recovery in response to therapeutic interventions. Now coming to the role of single fiber EMG in the follow-up of peripheral nerve injuries. So this single fiber EMG can be used to follow the course of reinnervation. So the parameters we should see are fiber density, mean jitter value, and percent blocking. Okay, all this we will see and determine the stage of reinnervation. Normally, the amplitude of action potential recorded with a single fiber EMG electrode from an average normal muscle fiber, okay, decreases to 200 microvolts when the electrode is approximately 300 micrometers from the muscle fiber. Thus, we can infer that action potentials which are greater than 200 microvolts arise from muscle fibers within 300 micrometers of the recording surface. By measuring many sites within a muscle, the mean number of action potentials that have an amplitude greater than 200 microvolts and rise time less than 300 microseconds can be used to calculate the fiber density. It quantifies the local concentration of muscle fibers within the motor unit. So normally there should be more than 200 microvolts amplitude, okay, within 300 microseconds. So that is normal. So if the time is more or if the amplitude is less, then we can imply that that muscle fiber is atrophied. Fibrillation analysis, EMG and dynamic echomyography in the follow-up of denervation atrophy and home-based functional electrical stimulation of a denervated or a reinnervating muscle. So the observations will confirm that home-based technique of FES and EMG and this echomyography, and they are useful for diagnosis and adjustment of treatments of denervated and reinnervating muscles. So in the later case, that is in the reinnervating muscle, dynamic echomyography provides information on the muscles or parts of the muscles contracting under voluntary act or under electrical stimulation. So they will give reliable information about the extent of the muscle of denervation and reinnervation, and they will give quantification of the progression of the atrophy, okay, to degeneration in a long standing muscle denervation, and what is the influence of electrical stimulation on muscle reinnervation. So they will provide all the information. In this manner, these techniques will contribute to the much needed evidence based approaches in physical medicine and rehabilitation. So in rehabilitation and physical medicine, we need evidence based approach. So this approach will be followed. Coming to summary, fibrillation potentials of skeletal muscle are small spontaneous action potentials which are considered to be a hallmark response of skeletal muscle to denervation. They have been shown to arise from both oscillations in the membrane potential and discrete membrane depolarizations resulting from changes in both sodium and potassium permeabilities. The fibrillations seem to prevent a more serious degree of initial atrophy in denervated muscles and are sometimes used to assess the degree of neural injury and reinnervation that have occurred. It is suggested that these may also be useful to assess the degree of atrophy that exists. For patients with peripheral nerve injury, therapeutic interventions such as electrical muscle stimulation may be helpful in preserving the contractility and extensibility of the denervated muscle tissue, and it will retard muscle atrophy also. But some literature highlights that it is a controversy on the electrical stimulation given to the muscle because it has some adverse adverse points also, but there are some benefits. So the benefits are more than the adverse effects. So the overall goal will be achieved, that is the muscle tissue will be healthier and viable until reinnervation is established. Monitoring fibrillations may be useful to verify that electrical stimulation is not detrimental to terminal nerve growth. That is, the stimulus parameters we have to wisely adjust and select, and the amplitude is applied should be at a low level. Okay, that means at a level that does not cause fatigue. Okay, does not cause fatigue, and the does not cause fatigue, the regenerating nerve, or stress the healing tissue. So the electrical stimulus should be as needed as that the stimulus parameters should be adjusted, so that the amplitude should be adjusted at a level that the muscle should not undergo into fatigue, or the healing tissue should not undergo into stress, or the regenerating nerve which is damaged, that should not be again damaged. So if you can see all these things, then electrical stimulation of the muscle is quite okay. So up to here we are learning about fibrillations and denervation hypersensitivity. So in denervation hypersensitivity, the fibrillations are common. Why these are appearing in a skeletal muscle fiber in case of denervation? We have seen all these things. So the main components are increase in the sodium conductance, decrease in the potassium conductance, increase in the acetylcholine receptors, and their distribution is also increased. So they are not only present on the motor end plate, but they are distributed over the entire skeletal muscle membrane surface, that is entire sarcolemma. And the calcium levels in the cell, they are increased. This calcium is coming from sarcoplasmic reticulum into the muscle, and the energy metabolism is also altered. The ATP is also decreased, so sodium potassium pump cannot function properly, so it is also leading to fibrillations. The next video is about abnormal muscle tone. So in this we have to know about uh, motor unit and the stretch reflex. So we have to discuss about stretch reflex in detail, in because stretch reflex will maintain the muscle tone. Then what are the abnormalities in the muscle tone, in which cases we encounter these abnormalities, we can see.